everyone, I'm Hannah.
And I'm Maya.
And today we are going to be diving into the fascinating topic of the PDGF signaling pathway.
Imagine you get a cut. That cut will trigger a positive feedback loop of blood clotting to close the wound and stop the bleeding. The initial stimulus is an injured blood vessel exposing collagen in damaged tissue.
Cells called platelets in the blood stick to the damaged site and release chemical signals, which attract more platelets to the site of the injury. The cycle continues, speeding up the clotting process until the clot is large enough to stop the bleeding, which acts as a signal to end the loop.
Now that we've covered how the positive feedback loop works generally, we can go into more detail about the PDGF signaling pathway. PDGF, or platelet-derived growth factor, is a protein released by platelets that plays a crucial role in the blood clotting process. It is hydrophilic and polar, meaning it is soluble in aqueous environments such as blood plasma, which allows it to travel through the body to its target cells. It is also dimeric, meaning that it is a monomer made of two subunits.
PDGFs are also in charge of sending out signals to increase macrophage activity during wound healing. PDGFR, or platelet-derived growth factor receptor, is a transmembrane receptor protein that has both an extracellular and intracellular domain. PDGFRs are crucial for wound healing and tissue repair because they stimulate the proliferation and migration of fibroblasts, which are essential for tissue regeneration and the maintenance of homeostasis after injury.
Now, there are some advantages and disadvantages to this pathway. The advantages of this pathway are that hydrophilic signaling is very rapid. PDGF stimulates cell growth and migration, which is essential for wound healing. And since overactivity drives diseases, PDGFR inhibitors can effectively treat specific cancers and fibrotic disorders.
And as for the disadvantages, because the response is so rapid, PDGF response can only go short distances. Overexpression or mutations in PDGF or PDGFRs can contribute to tumor growth and even cancer. Excessive signaling leads to severe fibrosis in organs like the liver, lungs, and kidneys.
So what causes the PDGF signaling pathway to begin? PDGFs are released from platelets immediately following tissue injury to initiate the healing process.
PDGFRs are activated when the PDGFs bind to the receptor's binding site. When the PDGFs form the ligand-receptor bond, they cause the PDGFRs to undergo dimerization. Dimerization for PDGFs is a process in which the two monomeric PDGFRs pair to form a dimer upon ligand binding, activating the signaling pathway. Once the PDGFRs dimerize, they activate themselves through a process called autophosphorylation. In this step, receptors transfer phosphate groups from ATP molecules onto themselves. The conversion of ATP to ADP during this process releases energy, which helps activate the receptor to initiate the response.
The three main responses to the PDGF signal are cell growth, directing cell movement, and changing the cell's shape. In the context of wound healing, PDGF signals to macrophages for the positive feedback loop associated with blood clotting, which speeds up wound healing. It can act as a paracrine or autocrine growth factor, signaling responsive cells such as fibroblasts in close proximity to proliferate. During PDGF stimulation, these cells are recruited to the wounded area. Fibroblasts migrate to the wound site, proliferate, and produce new extracellular matrix to repair damaged tissue.
All good things must come to an end, so eventually the PDGF signaling pathway must also. Once cell proliferation is no longer needed, or in our case, when the wound is fully healed, the PDGF ligands disassociate from the receptors. The receptors are thus dephosphorylated, simultaneously deactivating the receptors. This causes the PDGFRs to open up once again, which marks the end of the signaling pathway.
If you want to dive deeper into this subject, here are some links to articles we found riveting and chock full of information.
Thanks for watching, and we'll see you next time!